Welcome. We're going to do a little more detail on the nervous system today. We're going to go all the way down into the cell and look at what's going on and what it takes in order to get a nervous transmission to going through the nervous system. Okay, so here we have the membrane. So this is the neuron membrane. And it's separating the inside from the outside. On the outside of the cell, you're going to have different concentrations of ions. On the outside of the cell, you're going to have high concentrations of sodium. You're going to have high concentrations of chloride. And you're going to have low concentrations of potassium. On the inside of the cell membrane, you have different concentrations of ions. You have high concentrations of potassium. You have low concentrations of chloride. And you have low concentrations of sodium. As well, on the inside of the membrane, you have ions present. So these large anions are present within the cytoplasm of the neuron. Okay? And having these concentration differences leads to a membrane potential of about negative 70 millivolts. So this negative 70 millivolts is called the resting membrane potential. Okay, and these concentration gradients, as well as the resting membrane potential, is held in that range by two mechanisms. So the two mechanisms are going to be ion leakage channels and a sodium-potassium pump. And let's take a look at that within the textbook. So here is one of the ion leakage channels. These can be sodium or potassium ion leakage channels. And beside that is the sodium-potassium pump. And again, these two mechanisms are what are used to keep the resting potential at around this negative 70 millivolts. Okay, let's take a closer look at this sodium-potassium pump in the textbook here and exactly how that mechanism works. So this is the inside of the neuron and this is the outside of the neuron, and this is a receptor protein within that membrane. This receptor protein is attractive to sodium ions that are inside of the neuron. The sodium ions go into the receptor protein and bind. Then along comes ATP and phosphorylates the protein with its bound sodium, which causes a conformational change so that the sodium is sent out to the extracellular area, so outside of the membrane. Once this protein is open like this, it becomes attractive to potassium ions, and potassium ions come in and bind to sites within the receptor protein. At that point, the protein dephosphorylates and changes conformation once again to bring those potassium ions into the inside of the cell. And so this pump is running all the time, maintaining that negative 70 millivolts, that resting membrane potential. Okay? So from there, let's talk about the different potentials that actually lead to nerve impulses. So you can have a graded potential, and this is a transient change in the membrane potential, and it's due to chemically gated ion channels. Okay, and let's take a look at one of these chemically gated ion channels within the text. So here's a receptor protein within the membrane, and this is an ion channel that is closed until a neurotransmitter, a chemical within the extracellular fluid, comes in, binds to this receptor protein, which leads to the ion channel opening, and at that point you get an influx of ions, which is going to change the membrane potential, leading to a depolarization event. So a depolarization event is when the membrane becomes more positive. You can also have a hyperpolarization event. So depolarization and a hyperpolarization. So this is when the membrane becomes more positive and this more negative. Okay, these can also summate and make the polarization events more extreme, or they can negate each other. Okay? The other kind of potential is an action potential. And this is an all or nothing event. And it's triggered by a threshold potential. And within mammals, that's around negative 55 millivolts. Okay? And so once this negative 55 millivolts is reached, it's an all or nothing event. And these action potentials are governed by what are called voltage-gated channels. So when this is reached, these voltage-gated channels will open and you'll get a flood of ions coming in. And with that, let's take a look at what an action potential looks like. So normally the neuron membrane is at a resting potential here, again negative 70 millivolts. When you have an impulse that comes along that goes beyond that threshold potential of negative 55, you'll get these voltage-gated sodium channels opening, which will flood sodium into the cell. At some point, you'll get the opening of potassium channels and the closing of those sodium channels. So here's the sodium channels closing and the potassium channels opening. During the falling phase, you'll get the potassium channels also closing, but they close a little later than what is necessary, which leads to what is called an undershoot phase. So that's where you get this little undershot line here. And let's draw this particular graph to see this. So here's the membrane potential. In millivolts. And this is time in milliseconds. And here's -70, here's 0, and here's +55 or so. So on the rising curve, those sodium channels are opening and sodium is entering. You'll get potassium channels opening during the falling phase to reestablish that resting potential back down to -70. And again, those potassium channels stay open a little bit longer than necessary, which leads to this going a little bit more negative than the resting potential. And at some point, you want this process to end. And there's a couple of different ways that action potentials are terminated. Action potentials can be terminated by enzymatic cleavage. This enzymatic cleavage, one of the very good examples is acetylcholinesterase. It can also be terminated by reuptake by the neuron. And this is done via transporter proteins. And it can be, the neurotransmitters can be uptaken by adjacent glial cells. And with that, we'll conclude this section on nerve impulses and how they're transmitted throughout the nervous system.